to the juxtaglomerular cells. Okay, so that was the second mechanism that causes the release of renin. So, decreasing glomerular filtration rate results in decreased pressure, increased reabsorption of sodium ions from the proximal convoluted tubule, fewer sodium ions present in the filtrate of the distal tubule. Hence, the maculodensin cells respond by triggering the arteriole to dilate directly and releasing the prostaglandins, which result in the renin secretion. Okay? So going back to the earlier slide, this is the second reason that causes renin to be released. Okay? The detection. So maculodensin cells detect a decreased concentration of sodium ions in the filtrate. And that will cause prostaglandins to activate the juxtaglomerular cells to release renin. And then the third mechanism is a neurorenin mechanism. Yes?
It's going to sense the lack of the sodium ions inside the
distal tubule.
Okay, not inside the maculodensin.
No. So the maculodensin are essentially osmoreceptors. They have receptors on their surface that when they don't bind sodium, that activates them to release the prostaglandins. Okay? And so it's sensitive to the sodium ions right here as it's passing through the lumen of the distal tubule and flowing over the maculodensin.
Okay.
Okay? And then the third is a neurotransmitter control. All right, sympathetic neurons. There's actually very little autonomic innervation of the kidney. The sympathetic neurons will activate the afferent arterioles and efferent arterioles. They will bind to release neurotransmitters at the proximal convoluted tubule, and that's about it. Okay. Parasympathetic, I can't think of any role that parasympathetic has, certainly on the bladder, and the ureter going to the bladder, to cause the bladder to contract, and the ureter to have peristalsis moving the urine into the bladder, but not at the kidney itself. Okay? But the autonomic nervous system can be activated throughout the body, and certainly norepinephrine, epinephrine from the adrenal glands in the blood itself can cause the sympathetic neurons to release at the kidney site norepinephrine and to bind to beta-1 receptors. So sympathetic, direct sympathetic. So in other words, the smooth muscle cells, the JG cells, have beta-1 receptors on them. Okay? So those are the three different mechanisms that can trigger renin's release. Now, what does renin result in? Okay? Renin is an enzyme. So remember how prothrombin got converted to thrombin when we were talking about coagulation? And thrombin converted polymer round globules of fibrinogen, strung them together into long fibers of fibrin. Okay, so the thrombin was itself an enzyme that caused that change to occur. Renin is an enzyme, but instead of stringing long proteins together, it cleaves off. It removes the amino acids from a protein to activate it. All right. So again, if I can erase this portion right here, we'll look at the effect of renin. You guys done with these three steps? Okay, they're the ones essentially that were on the board. So now renin is released for any one of these reasons. Renin is released into the blood, all right? And it is an enzyme. This is not the renin from calf stomachs that people form gelatin or junket or stuff like that. So it's spelled with 2 N's. This is a different enzyme. And it works on a protein known as angiotensinogen. Angio referring to vessel, and tensin referring to pressure. Okay, when something's tense. So it converts angiotensinogen to another molecule known as angiotensin 1. There are a few other cells that might make angiotensinogen, but primarily it comes from the liver. Angiotensin 1 is an intermediate stage. Okay? It by itself has a slight function, but not much. And so it needs to be further converted to angiotensin 2. Actually, I don't want that long an arrow. You guys in the back won't be able to read it. Now, even though this first reaction takes place in the kidney, the second reaction takes place in the lungs. And the enzyme is called ACE, which stands for angiotensin converting enzyme. And that converts it to angiotensin 2. Angiotensin 2 has 2 functions. So we're at this point right here. Renin converts angiotensin, and eventually we end up with angiotensin 2. 2 functions. By itself, it will cause systemic arterioles to constrict. Like we already said a few minutes ago, that increases peripheral resistance and blood pressure goes up. Okay, so angiotensin 2 can do that all by itself. Can I erase this right here and move this up so you guys can see it in the back? Does everybody have that down? Okay. So it will cause constriction. Of peripheral arterioles, including the efferent arterioles. Not the afferent, but the efferent. All right, this increases blood pressure by increasing peripheral resistance. And its other function is it causes the adrenal cortex to release aldosterone. This is where we came in with the heart. So aldosterone is a hormone. It's going to get into the kidney via the adrenal arteries and go through all of this structure. And then on Tuesday, we'll see more specifically how it works. It actually is a steroid hormone, right? And it's going to cause distal convoluted tubules to reabsorb sodium. Now you might say, well, all that's going to do is make our sodium ions even lower, and that's what triggered the whole thing to begin with. But remember, when these sodium ions are reabsorbed, what's going to follow passively? Water. Okay? So water is reabsorbed as well. And systemic blood pressure goes up. as a result of increased venous return, increased stroke volume, increased cardiac output. Okay? So this is a more long-term, takes a little while for this to occur because aldosterone is a steroid hormone. All right? It has to actually cause the synthesis of additional sodium ion pumps to return that sodium. Now, in addition, the macula densa cells, okay, will cause the afferent arterioles to be dilated. They release a chemical that inhibits the activity of the smooth muscle and causes the afferent arterioles to be dilated and increase the blood flow into the glomeruli. So this is how the kidney tries to maintain some control over that filtration pressure. Okay? Obviously, if filtration pressure is too high, we can damage the filtration membrane. So if systemic pressure is high, all of this stops, and we actually have constriction of the afferent arterioles and some dilation of the efferent to lower the pressure across the glomerular capillaries. Okay? So this is that first step in filtration. So, and how it's regulated. So now we have the water, we have the small particles, we have ions, we have glucose, we have amino acids, we have urea and ammonia and creatinine, and we have those particles now all forming the filtrate. So the rest of the process that happens in our distal and proximal convoluted tubules is the reabsorption, which is the primary activity, and the secretion, adding additional components to it. So let's look at that before we take a break. And we're going to look at proximal convoluted tubules, right? That's the next structure. Proximal means closest to the point of origin. And the point of origin is our capsule. So on the models, that's how you can identify it. If the tubule is attached to the capsule, it's the proximal convoluted tubule. Now, histology, microscope histology is not on your quiz today. It'll be on Thursday's quiz. But we're going to look at some images of that. So when we look at a cross-section of the proximal convoluted tubule, it is formed of simple cuboidal cells. But they have very long microvilli. It gives it a very messy appearance. Microvilli have actin in them. They're not motile. It functions to increase surface area. And that, of course, is for our purpose of reabsorption and to some degree secretion. Distal convoluted tubules also have simple cuboidal cells. The nuclei are very close to the surface, so they kind of bulge out a bit into the lumen. Now, even though I've drawn the lateral membranes in the microscope, they're hard to see because they fold back and forth so many times. We do have microvilli, but these are short. Not as much reabsorption or secretion is taking place here. Okay. And so we'll see this when we actually look at the stained histology of the slides. Now, we're going to take a couple of these simple cuboidal cells from the proximal convoluted tubule. And this side is going to be the lumen. So this is where we have the filtrate. And that's the side that the microvilli face. The lateral and the bottom membranes are wavy, but they're not thrown into microvilli, so there's a slight increase in their surface area. And I'm not going to put in the nucleus, all right? Then over here, we would have the blood vessel. And this would be the peritubular capillary. Okay? So I'm just kind of redrawing what's on the board. Now, reabsorption refers to the direction of the movement of particles, all right? So when particles move from the filtrate and go back into the peritubular blood, now we're going to have, in our distal tubule or even our nephron loop, we're going to have less of these particles in the filtrate than we do in the blood, because we took them out of the filtrate and we added them back to the blood, okay? So that direction of movement from filtrate to blood is known as reabsorption. We're reabsorbing. That's the whole purpose of the peritubular capillary, is to pick up this material that's being reabsorbed back into the blood. The arterial flow through the kidneys is multiple times a day, so that approximately 180 liters of filtrate are formed. That's way more blood than you actually have in your body, which is about 5 liters, okay? So obviously this is formed over and over and over, and you reabsorb it. Otherwise you'd be sitting on a toilet with an IV hooked up to your arm, okay? Or else walking around with, 15 or 20 Foley catheter bags attached to you. So we have to reabsorb. It's about 2 liters are actually excreted as urine. Okay? So there has to be reabsorption. And this is the whole process of the kidneys in this filtration. Now, we're going to kind of start at this end. Okay? A lot of the movement of the particles from the filtrate to the peritubular capillary are done in conjunction with the movement of sodium ions. Remember I said 80% of the sodium ions are returned to the blood from the proximal convoluted tubule? Well, sodium ions are going to naturally diffuse into the cell. Remember they're in lower concentration inside a cell than they are in the blood or in extracellular fluid. To get them from the cell into the tissue, we have to use energy. All right? We have to use active transport. So we'll draw in a protein here, which is our active transport pump. And it's going to move, the other way around. It's going to move sodium ions out of the cell, and it's going to move potassium ions into the cell. You remember the sodium-potassium pump, 3 and 2, all right? Well, the potassium wants to get out anyway, so we end up, to keep us from having too many potassium ions in the cell, the potassium just goes out a leaky channel. And it just diffuses right back out. But we are removing sodium. So what are we doing to the amount of sodium in the cell versus the amount of sodium in our filtrate? We're increasing the gap between those two levels. All right? What does that do to diffusion rate? If we go from a difference like this to a difference like this, it speeds it up. It increases the diffusion rate. All right? So this is how we take advantage of this. This is known as active transport, right? Because we're going against the concentration gradient. We're going from low to high instead of high to low. It uses ATP to run the pump. So guess what organelles are stuffed at the bottom of the kidneys. Tons and tons of mitochondria to provide that energy. So M stands for mitochondria before it gets too busy and messy here. So we're pumping sodium out, which means the inside of the cell is getting very low in sodium. So the sodium now that's in our filtrate has a much higher drive to go into the cell. It's an ion. It's not going to just cross the plasma membrane. It has to go through a protein channel. So that protein channel has a place that hooks up with sodium to allow it to pass through. It also has a place that hooks up with glucose. Because we have glucose particles are small enough to diffuse into our filtrate. They normally require a transport protein anyway, because they're too big to cross the plasma membrane. So in this case, that transport protein is hooked up to sodium. And we increase the amount of glucose removed from the filtrate by this association. Now this is known as a secondary active transport. So sodium is moving into the cell down its concentration gradient. Glucose is moving into the cell down its concentration gradient. So the two are moving in the same direction. Sodium and potassium were moving in opposite directions. So this is known as an antiport. This protein molecule is an antiport because it's transporting the two substances in the opposite directions. This molecule is known as a symport. Two molecules are going in the same direction. And this is known as secondary active transport. Now I touched on this briefly in 430. I don't think any of the students that had me in 430 that are in my morning class remembered it. So you may not remember it from your 4 to 30 class. It's discussed with membrane molecules in the chapter on plasma membrane, okay, and transport, but you probably don't remember it. Does it require energy at this particular location right here? No. So secondary active transport is really passive diffusion linked to the active transport. It occurs, or it occurs at a faster rate due to the energy expended to decrease the amount of sodium in the cell. All right? But secondary active transport is really a form of diffusion. Sodium and glucose are both going from their area of high concentration to their area of low concentration. Okay? Once the glucose gets in the cell, and I fill that cell up, so once the glucose gets in the cell, it actually leaves via a facilitated diffusion. Which is a form of diffusion, but it requires a protein molecule to enable it to cross the plasma membrane. Okay. Now, glucose isn't the only molecule that uses secondary active transport. As you can see over here on the graph, amino acids will be moved across the membrane at a faster rate. Certain ions, certain vitamins will move across at a faster rate as well because of that. So from the filtrate of the proximal convoluted tubule, okay, if we've just done our filtration, it's normal to have lots of glucose to the level that we have in the blood, lots of amino acids, but those are not normal components of urine. Once we are past the proximal convoluted tubule, normal filtrate will not have amino acids or glucose. We don't have symport proteins to remove amino acids and glucose except in the proximal convoluted tubule. Okay? So if someone has diabetes mellitus and their blood sugar levels are elevated above normal, there are insufficient numbers of glucose secondary transporters to move the glucose out of the filtrate. And glucose will stay in the blood. Will stay in the filtrate. Okay? And that's called spilling over into the urine. That's what mellitus means, it's sweet. So diabetes mellitus is a polyuria. It's a high level of urine production. If you were to dip your finger into it, remember urine is sterile. At least until it leaves the body, it passes through the urethra, it's sterile. It will taste sweet. As opposed to diabetes insipidus, which means bland tasting. And we'll talk about that on Thursday. It has to do with levels of ADH, which both of them increases the volume of urine. Okay? So what would be the effect of having additional glucose molecules in the filtrate on the urine volume? Remember what happened to strawberries when we added sugar to the strawberries? Pulls water out of the strawberry cells. So glucose in the filtrate also has an osmotic property. If the glucose stays in the filtrate, it's going to keep water molecules. Hence the symptom of polyuria, increased urine volume if someone has diabetes mellitus. Okay? Now let's look at something slightly different. Remember what potassium cyanide did? We talked about this briefly when we talked about forms of hypoxia. And potassium cyanide works at the mitochondrial level, and it inactivates the enzyme, one of the cytochrome oxidases that allows the mitochondria to use oxygen in the very last step in the making of ATP. So if a person is poisoned with cyanide, it affects their mitochondria and they can't make ATP. So let's ignore the effects in all the rest of the body. If these mitochondria are poisoned by cyanide and they can't make ATP, what's going to happen to this process? What's going to happen to the level of sodium ions and glucose in the filtrate? Both of them will increase, all right? Because we won't have this sodium pump working, because we won't have the energy to run it. Without that working, sodium's going to be higher inside the cell. If sodium's higher inside the cell, we're not going to have the strong drive with our symport channel to move sodium into the cell. And without that happening, both sodium and glucose and amino acids will remain in the filtrate. Okay? And both of those will function to increase urinary volume because they'll both have an osmotic effect. All right, other molecules, some of our urea that we're trying to get rid of will still passively diffuse across. Water will follow. We have aquaporins. We won't really talk about them till Thursday, but there are water channels because water is not soluble in the lipid of our plasma membrane. So aquaporins will allow water to also follow the osmotic gradient of the sodium and the glucose moving across the cell. Chloride ions, several other ions will actually pass between the cell. What do you think is causing the chloride ion to move? That electrical charge. It's a negative charge, sodium's a positive charge. So as we're moving those positive charges out of the cell, chloride's going to follow because of its negative component. Okay? So by the time where the filtrate has passed through the proximal convoluted tubule, in a normal situation, 80% of the sodium ions should have been removed. % of the chloride ions too, for that matter. 100% of glucose. 100% of amino acids. So the rest of the filtrate should not have any amino acids or glucose in them. Unless, of course, you've got really, really high levels, and then some of those will remain because we have insufficient transport proteins. Now, just before break, I want to define secretion, but we'll look at examples of secretion when we do acid-base balance on Tuesday of next week, okay? So I'm just going to show you the slide rather than redraw it. So don't worry about all this going on in here. We'll talk about that next week. But the direction, secretion is in the opposite direction, all right? So at the top of your lecture notes talking about secretion on page 22, draw an arrow going from the blood back to the filtrate. Okay? So secretion is going to be in this direction. We're going to take material that is still in the blood that was not filtered, and we're going to add it to the filtrate. So you saw how with amino acids, when we reabsorbed it, we ended up with filtrate that didn't have any amino acids. Certainly our blood still does. With secretion, some of these particles might have been filtered already, and we have some of them in the filtrate. But with secretion, what we do is we take the particles that didn't get filtered, and we add additional ones to the filtrate. So we end up with even more in the filtrate. Typically what gets filtered are hydrogen ions and the products of drug breakdown. So antibiotics, one of the reasons why you have to take antibiotics so frequently. All right, usually there's a few that you can take once a day. But usually it's every 4 to 6 hours, sometimes every 12 hours. They get broken down and metabolized and excreted in the urine. Some of them even to make your urine look orange. Okay. Something that is actively secreted is that chemical from asparagus. Some people do that, not everybody does. Okay. We add to it. So I just want you to understand at this point in time, secretion is in the opposite direction. We end up with more of that component. All right, so let's stop here, take a break, and then we'll come back for just about a 20-25-minute discussion of the function of the nephron loop, and then we'll go over that all over again, the nephron loop on Thursday.